We'll start with the next. We'll start with the next session for today. I would like to call upon the stage chairpersons for this session, Dr. Subrat Kumar Mohanty. Please come on stage. Dr. Vivekanand Jindal, our first speaker for this session is Mr. Sumant Bhutoria. He's the CEO of Alpha Tech Systems, CEO and founder of Alpha Tech Systems.
Hello, can you hear me at the back? All right, thank you. Should we start? Start. Right. So a warm welcome to all the participants here. My name is Sumant Bhutoria. I'm the CEO of Alpha Tech System in the 3D print for about 12 years now, and working in a wonderful field. A lot of customers all over India have now start my slide. So basically, a little bit about my company. Been doing this for about 13 years. Our main business is 3D printing. Build customized 3D printers, robotics, industrial automation, medical automation, drones, IoT, etc. Uh, a little bit about myself. I did my BTech electronics from IIT Kharagpur, then a master's from Arizona State, from Ahmedabad, and then I started my company Alpha Tech. We have built up a presence in more than 250 R&D labs all over India. A rich client base, New Delhi, ISC, IIT, Tata Cancer Hospital, then within pharma companies, Alchem Labs, Zim Labs, Manipal. We've got a good share of recognitions, achievements in India, papers, and so on. We've been invited by the Chinese government well as a special speaker to talk to them, talk to their makers about times of CNBC. These are our 3D printer offerings. We manufacture our Anga range of bioprinters. They're also authorized dealers for various reputed European companies. So, Research, Printer, Region, B9 Creations, Frozen, and then for BioWings, so on, we've tied up with UPM, Manchester Biogel, Tata Steel. We also work closely with some of the universities like IIT, Chitra, SCM, Trivandrum, or various BioInk commercializations. These are our various 3D printer service offerings. People who don't want to buy a 3D printer, they can come to our place and can provide 3D service to them. Uh, let's start with the presentation today of why we need 3D printing in medicine and healthcare. First of all, pointer. Hold on. Okay, good. Uh, talk about patient-specific implants. The patient can customize the implants and implant them rather than just relying on the implants. For surgical planning, VR headsets, etc. You can 3D print the affected. Not necessary to have. You can print them out and use them in your workshops and so on. Then for doing surgical tool guides, we had a number of cases with hospitals where they wanted to print guides during both the operation as well as post recovery. Lot in prosthetics, orthotics, There's the idea of a 3D printed hand, which is a low cost device, particularly useful for pediatricians because children, when they want to have these kind of prosthetic limbs, because children are growing, so prosthetics will tend to become small after as they start to grow. Low cost, it's much easier for them to rely on these low cost prosthetics. This can be done easily with 3D printed. Finally, various types of custom medical devices, etc. For prototyping purpose, 3D printing is very useful. Brief overview of the different types of 3D printing technologies. So you will hear about selective laser sintering, which is normally used with metals, polymers, and so on. Then stereolithography or DLP, which is again very popular. We use a liquid curable polymer and use this to create very fine structures. Uh, then finally, fused filament fabrication or FDM. This is the most common method used in 3D printing. We use thermoplastics. It is one of the cheapest ways of 3D printing. The machine costs are also fallen very dramatically now. You can get very reasonably priced machines for as low as about 1 or 2 lakhs. The cheapest ones are as low as 20,000. Then, of course, bioprinting. So we'll talk more about this in detail in next slides. FDM. So here I will talk a little bit about the process. So here the way it works is first we do a 3D design. Then we use software to slice this design into various layers. Okay, so you see this is the shape and then we digitized or we sliced it into different layers. And then the computer will generate the G codes. This directs the machine to deposit the path for each layer. Then finally, layer by layer by layer, you build up the entire structure that you have this as a final object. The various constituents of a 3D printer, so the nozzle, the heat bed, the linear positioning, accuracy, and so on. And I'm going to skip this because we have a paucity of time. We've done various, measurements on the quality of the FDM prints, etc., under microscope and so on. You can see good resolution, etc. These are some of our work that we've showcased for various medical applications. So, uh, maxillofacial region, the spinal region, uh, the orthopedic applications. These are prints that are used for dentistry, so dental guides, plants, molds, and so on. Now, coming to 3D bioprinting, which is a more specialized topic. So, why do we need 3D bioprinting? Need for 3D bioprinting is simple. The clinical case where you want to use it for tissue engineering or organ fabrication. So the big idea here is that instead of having to rely on third-party implants, we can take living cells from our own body and 3D print into the required organ or tissue patch and then implant it back to our body. So the idea here that since I don't need a third party donor, I don't need to suppress my immune system. So the quality of my life post operation is similar to what it was before because my immune system remains as strong as before. I don't need to suppress it. And of course, the problem of not having to deal with finding third party organs and so on. For the pharma case, 3D bioprinting is very useful because the 3D printed tissues are used for drug discovery purpose, for personalized medicine or 3D printed tablets, and then finally in the skin care, cosmetics for hair and the fairness of skins and so on and so forth. This is the workflow for 3D bioprinting in very simple terms. So you start the CT MRI scans, then you use it in 3D bioprinters, various types of 3D bioprinters, and then print it into the particular tissue patch or organ that you would like to do. This is the comprehensive workflow for medical healthcare. So we start with CT MRI scans. First stage, you can use the traditional 3D printers for pre-surgical modeling. And then finally, the 3D bioprinters, which will take living tissue and so on, and then use it for growing into the tissue patch. At this point in time, we've not been able to do very complex organs or so on. Simply put, we've been able to do liver, heart, tissue patches. And in the case of skin, again, we've had some success there. terms of modeling the keratinocytes, fibroblasts, and the dermis epidermis. And then finally, after the 3D bioprinting is done, we will incubate it. And after it has grown to the particular size we want, we'll implant it back into the animal for post cure studies and so on. These are some of our partners for 3D bioprinters. So my own company will make the low-cost 3D bioprinters, and European partners, Printer, Ready 3D, Religenu, for various volumetric and extrusion-based bioprinters. For bioinks, again, we make the basic bioinks ourselves, and we've partnered with various companies, Manchester Biogel, UPN, Sri Chitra and Trivandrum, and Tata Steel for various bioinks. This is how our bioprinter looks like. So apart from our bioprinter, we've also worked to create a plethora of accessories and other equipment that may be needed to work in bioprinting. Now we talk a little bit about personalized medicine or 3D printed tablets. So here, what is personalized medicine? In simple terms, it is a therapy with the right drug at the right dose given to the right patient. Pretty similar to what we had in the olden days of the Hakim, where they used to make the ointments very specifically as per requirement of the patient. So modern medicine, of course, relying on the large scale volume pharma, where it's not possible to customize each and every dose. The idea of 3D printed tablets comes in, where each dose can be customized as per the requirement of the patient. And also you can fabricate the dose in such a fashion that it's meted out to the patient at regular intervals. So if you want an intermittent kind of dosage, or if you want a delayed dosage, or if you want the dose to get affected once it reaches a particular region, all that becomes possible with the use of 3D bioprinted tablets. Okay. Uh, Moving forward, this is the world's first 3D printed tablet, a company in the US, Spritam, which got a US FDA approval as well. And this is work that we've done in India for a pharma company called Zim Labs in Nagpur, where we've integrated the 3D printing into their pilot production machines for doing pilot line production of various thin film based tablets. Various other 3D printing techniques. So inkjet dispensing, this is something that we found a lot of interest from various pharmacologists as well as doctors, where they wanted a cheap equipment that can do micro dispensing of liquids. So we're able to use our own bioprinters to dispense droplets at less than 1 millimeter kind of spacing. We've also integrated now microvalve dispensers from Switzerland into our printer. So this gives even higher accuracy for people who want to use it. This is examples of 3D printing with bioceramics. So we worked with hydroxyapatite, zirconia, TCP, and so on to make various soft bone tissues, so on. This is work on electrospinning. So again, in our machine, we have electrospun various bioinks such as PCL or PVA and so on, spin them into very fine mats, which can then be used for various purposes, applications. One major application of electrospinning in bioprinting is to create the extracellular matrix, ECM, on which the final living cells can rest. So the ECM acts like the protein structure, allowing the living cell to find its more natural environment. And in order to do that, we've used electrospinning within our bioprinters to produce that. I think we are the first in India to do that, probably in Asia as well, to have this powerful capability at a fraction of the cost of what is charged by the European manufacturers. This is some slides on 3D printed silicone, orthotics, medical implants, so on. So 3D printed soles is now becoming fairly popular. So you will scan the patient sole and then create 3D printed customized soles. People have added an extra layer of adding some electronics or sensors to the foots so that the patient's movements can be monitored, recorded at specific periods of time. So all that becomes much simpler with the use of 3D printing technologies. And then finally, I come to the end of my slide. Just as a conclusion, 3D printing, what it allows you to do is to allows you to do what you have in your mind at a very fast pace. We are talking about a few days, sometimes even hours. Is it something simple? So the development time, the prototyping time, the time to it allows you to customize your products as per the requirements of the patient. And finally, all this possible at a fraction of the cost had we had to rely on traditional technologies. All right. So thank you so much for hearing my presentation. If you have any questions or if you would like to contact me, those are my details. So you can note them down. You can reach out to me for any of your requirements, and I'd love to hear back from all of you. Thank you.
Thank you very much. Any questions, please? We have 5 minutes. We have 5 minutes. No?
Yeah.
Yeah.
Dr. Sumant,
Yes.
yeah, I am chair.
So,
Excellent, I think innovation. We have a multicolor 3D printing in this so
yes.
that we can have a artery vein and the tumor supposing and doing. So that is in vitro simulations can be done. Then it can be then try to minimize the complication in the surgery.
Yes, very much, sir. In fact, we've installed a similar printer at Tata Cancer Hospital in Varanasi. So it's a 5 tool head printer, which allows you to work with 5 different multi-materials or 5 different colors if you want of the same material. The bigger challenge in these areas is basically identifying the soft tissues, segregating them out, and then creating the workflow for the printing of these 5 different colors. So the majority of the effort goes in towards identifying the soft tissues, because you start out from the MRI or CT scans, and then you have to identify the regions where you'd like to have different muscles, tissues, or different medical arteries and so on and so forth. So that takes up a lot of time, especially if you are dealing with soft tissues like say liver or a heart or so on. So on a patient-to-patient basis, it becomes a little more costly or more complex. But of course, for generalized kind of studies where prior work has already been done, it's pretty simple. I mean, the files are all there. We just need to print them out. So that's pretty trivial. The main challenge is basically in conversion of the patient-specific MRI CT scan data into the particular file formats that you. That takes up time.
And about that, a lot of time the consistency of the tumor also varies.
Yes.
Like a tumor is very soft or a hard tumor, we don't.
Right.
And how the tissue will, but will not mimic that, I hope.
Yes, so I mean, we'll have to work with the doctors to identify which are the areas for soft and hard. So until we know, we can't really model it in the software. So that is the big challenge.
That will be a challenge. Yes.
Yeah, yeah. And there are very few specialized people who are trained to do this. So that also becomes a big challenge to find the kind of people. So in fact, I'll tell you about our own company. We had one designer who was specialized in doing this, and then he left us. So it's difficult now for us to find a replacement. So I know how to do it, but I'm not a specialist in that. So till the time that we can get another replacement who's trained enough, or we need to train him, he has to spend time to learn all that. It becomes a challenge for us to kind of do this work. But yes, I mean, there are people, I think there are specialized companies who are doing this, but not so much in India. I think in the Europe and US, it's much more frequent, much more common. India, probably not so much at this stage. And I think also on a per patient basis, there is not so much immediate requirement. As a generalized, yes, I mean, there are models which can identify and they can.
Cost may be a hindrance factor. Yeah,
Yes, cost is an hindrance because the development cost that takes up a fair chunk of.
Thank you, sir. Excellent
Most
talk.
of. Thank you.
Thank you, sir, for this enlightening talk and for enriching our scientific session. For the next talk, I would like to call upon stage Dr. Ninad. He is currently working as an assistant professor in radiation oncology in TMH Varanasi.
Good afternoon, everyone. Thank you for the opportunity. So thank you for the kind introduction. I'm Dr. Ninad Patel. I'm Assistant Professor Radiation Oncology in TMC Varanasi. No financial disclosures, although a big disclosure, I am not a surgeon, I am not a pediatrician. Although my experience has been in setting up the 3D printing lab in our hospital for radiation oncology, but then inspired by us, surgeons have been taking our help for surgical planning. We have moved from 3D printing per se to direct virtual planning. So skip the actual printing step, since sir said there are a few intricacies that you need to master for that. So overview. So my topic is virtual surgical planning and 3D printing. So why does it matter and where does it help? What are the upfront challenges specific to pediatric surgery? What is the end-to-end workflow? So it seems like a very mystical thing to do 3D printing and reach the final product, which might look beautiful. You don't know how you got there. So what is the end-to-end workflow? Material selection. So like we've pointed out, multicolor, multi-texture, multi-softness materials. If we are thinking about permanent implants or even prosthesis, we need to talk about sterilization, biocompatibility. When you enter 3D printing, you are basically entering medical device manufacturing, and now you are competing with the big shots. So you need to know what are the regulatory minutiae, and we'll see a short adoption pathway. If you go home after the conference, how can you reach a level where you generate your first print? Why does it matter? So it has a steep learning curve, and during the learning curve, you will fall a few times. You will slow your workflow, but after you have passed the learning curve, it is going to shorten your surgeries. So obviously, you have all the advantage of short surgeries. You have less blood loss, reduced anesthesia risk, fewer post-op morbidities. Intra-op, you have fewer surprises. You already know what is the anatomy in 3D before you enter the OT. You are going to get better margins between the resection volumes and the normal part of the tissue that you want to leave back. Fewer reops as a result. Overall, although you might think it has a high cost to entry, if you see the cost of the morbidities that you're avoiding, it is overall cost neutral or cost saving. Where does it help the most? So in pediatric surgery, there are a few limitations. Although in surgery overall, complex bony resection or reconstruction, soft tissue function sparing resection is the main domain. In pediatrics, since we are dealing with growing bones, growing physes, we have a few limitations in surgical implants. One of the major advantages consenting. So it helps a lot to show the patient, this is your anatomy, this is the tumor that we're going to resect, this is the part that will be left behind, this might be the morbidity, we might have to excise this much extra. So this need not be printed actually, you can show it on the screen also. So that is the virtual planning. So if you actually print, then all of these advantages, plus you can end up with patient-specific surgical tools, either guides or implants or prosthesis. So what are the challenges? We already discussed growth and remodeling. So you have to think about future growth vectors and physes and avoid any implant that bridges physes and growth plates. Or I think the next speaker is going to talk about resorbable materials. So you can implant things into the growth plates which are going to be resorbed over a period of time. If not, then you have to anticipate symmetry drift as the contralateral side grows. So pediatrics is a soft tissue dominant virtual planning and printing domain, where we are concerned more about function preservation, as much soft tissue sparing as possible. Family communication and consent. One major concern in pediatric surgery is pediatric surgical planning is we want to minimize the amount of radiologic exposure, CT scans, X-rays, etc., that are required to capture the data for 3D planning. So we'll see the end-to-end workflow. What is the data in? So very rarely we have seen scenarios where we 3D scan the patient with a professional 3D scanner or a camera. Mostly it is DECOM data from PACS from the diagnostic imaging. If you have multimodal images, if you have a CT and MRI, registering them together. And the next is the task that we were discussing, structure segmentation. So this is something that surgeons are not used to, even radiologists also not mostly used to. This is our bread and butter basically. Radiation oncologists have been dealing with tumors deep-seated. surrounded by normal structures. We want to treat the tumor with a multiplicity of hundreds of beams from various directions, avoiding normal structures. So we need to know internally क्या कहा है और किसको avoid करना है. So those things require you to tell the computer कि this is where my tumor is exactly, precisely with a margin knowledge that is similar to a surgeon. We need to know planes internally. We need to draw normal structures to tell the computer कि ये इधर है. Now you can simulate the radiation beam. So this has been our bread and butter, or part of our bread and butter for the last several decades. So structure segmentation is something where we excel. You can take your help of a radiation oncologist if you want to enter structure segmentation. Soft tissue segmentation is not that difficult as you think. The next step is once you've generated this slice by slice segmentation, basically it is like in a heart, let's say. So drawing on it where your left atrium, right atrium, left ventricle, right ventricle is, arch of aorta, ये सब draw कर दिया. So this segmentation होने के बाद the software itself converts this slice by slice drawing into a 3D volume. And now comes the role of virtual surgical planning, where you in silico, you in the software, in the computer, you manipulate the 3D volumes. So we'll come to the example workflows if we have time. We'll skip it for now. But you can imagine that a multidisciplinary team, surgeon, radiologist, the OT technician who is going to design the prosthesis or implants, all of them sit together, they go through the 3D model, they decide what planes to take, how to repair the defect that is generated. So that is all virtual planning. The next step, if you are having the ability to 3D print, you print either the anatomy for planning to handle the anatomy, or you can actually print the surgical tools. 3D printing has a few more steps beyond the actual acquiring the actual print. You need to do QA for the print. So what you're printing, you're assuming you're going to find it in the body, may not be. So if your workflow is suboptimal, you're not going to end up with something that is accurate. So this is we'll come to this software specifically. So you need to go through a few steps before you end up with the print. This software is free open source software where you get to simulate and it generates the 3D models. So what do you do with these models? You can either just manipulate them in the software, or there are dedicated virtual surgery simulators. You can manipulate the model with very simple tools like turn off the tumor, turn off the kidney. In this case, let's say you want to see the feeder vessel of the tumor. So you turn off everything except the arterial supply. You see the venous drainage. You want to see how you are going to reconstruct the pelvic ileal system. So you turn off everything else. That is the simplest form of manipulation. But if you have a robotic suit, or you have something in between, like this is a Oculus Rift and the Oculus Rift controllers. You can manipulate these structures as if you're doing the surgery virtually, or the ultimate is going to be something like this. This is a commercial product where you are in the OT suit. All the OT equipment is pre-modeled in a library in the software. Whatever tools you have accessible in the OT are accessible in the software. You can actually perform a pseudo procedure with haptic feedback, with force feedback, as if you are getting some kind of chance at doing the actual procedure. So this is a simple procedure that we did. We have three procedures that we're going to show as a case capsule. This is a 2-year post-surgery mandibular reconstruction for a alveolar cancer. Dental wanted to implant teeth, permanent teeth into this. So we did a 3D model. Simple procedure, printed as many copies as they wanted. And then they got to actually do the implant into the physical copy, troubleshoot the process before they entered the OT for the first time itself. In the absence of this, the first time they would see the bone would be when they go into the OT and they start the procedure. So what is an easy and free starting point? There are paid commercial software available, but before that, I would urge everyone to start off with free open source software. So 3D Slicer is the one that we use. Free open source software, extendable imaging platform, where it is basically a radiology suite, but every specialty has its own extension in this. So if you see, neuro will have stereotactic procedures ka extension. Hepatobiliary will have liver segmentation, AI segmentation. So you don't need to manually segment everything. Some things are AI also right now. With extensive support and funding, NIH is funding it. Basically universities are funding this project. It is definitely not for clinical use. But if you have the ability to do the QA, internal commissioning and validation of the process, then the output you can use clinically. So basically, you're becoming a medical device manufacturer in that case at a smaller scale. So this is the software. If you have used Paint, most of you must have. You can recognize the simple tools. There is a Paint, Draw, Erase, etc. And there are more advanced tools like AI segmentation of bone, AI segmentation of lung segments, liver segments. So whatever you can have a huge data set for can be converted to AI. These are the way the extensions look. So individual extensions like here there's a resection planner for liver, heart, there's stereotaxy, there's bone reconstruction planner, ablation planner. So individual specialties have collaborated with individual engineering teams. We have collaborations with IIT, BHU. You can have collaborations with any software engineer if you want to develop your own extensions. The other side is so that was not intended for clinical use. This is purely FDA approved and commercialized. You're going to pay a heavy amount to them, but you're going to end up with a ready end product. So you send them the patient scan, you end up with something to hold in your hand. So you can see, I'm not going through all of them, but you're going to have multiple options in various domains. So once you've ended up with your virtual model, then comes the role of 3D printing. Like Mr. Sumant alluded to, there are multiple technologies. The simplest printers. So I began my journey out of boredom during COVID, and I got my first printer for 30,000 rupees. Right now, you can get the same thing or the latest version of the same thing for 12,000, 14,000. It will generate simple rigid plastic monochrome single material, which is still useful. So guides, tools, crude anatomic models, etc. So that jaw that I showed could be done on this also. You can get multicolor versions of the above. You can get multiple hardness and multicolor. But what I felt is there are no true substitutes to tissue. So if you see breast implants, you see your earlobe consistency. All of these are consistencies you can't get in 3D printing as such, but still some levels of hardness. So something like silicone, hard silicone, like a shoe ka sole, rubber tire, that level of flexibility all the way to rigid bone. Next, so this will be available at 2 to 3 lakh rupees. Next step above is when you go into the next decimal point. So metal ceramic implant grade printing going to be somewhere around 50 lakh to 2 to 3 crore. And then the fork is bioprinting, which is distinct from all of this. So what printer do you select? What material do you select is dictated by what endpoint you desire. This is our printer. So we have that printer which we spoke about in the previous talk, that 5 two-LED version. It can have 5 colors. Actually, the present market, you can actually use an MMU to give 5 more colors to each two-LED. So 25 colors, for example. That is not the, colors is not the specialty. You can use multi-materials, multiple hardnesses in a single model. So what is the actual process of that 3D printing? You start with segmentation, same as virtual planning. You convert the segmented scan into a 3D volume, same still in 3D slicer. Next, you optimize it for printing. So if you have additional screws, plates, guides, etc., you incorporate it into the model. Next, you slice it for printing, which is basically a software which converts that file model into a 3D printable file. And then you print. Printing minutiae are variable and they vary as per the printer technology. Printing, if you dream of starting it yourself, is it what your model, is it what you get finally after printing? No, you will get that structure with a lot of support structure with a rough texture, etc. So there is a significant amount of post processing required if you want something that exactly emulates human anatomy. Your output needs to be quality assured in case you are not going with a commercial solution. If in case you are using it intra-op, it is likely to touch tissue, then you need to go through the sterilization and QA of sterilization separately. Sterilizability of individual materials and techniques is different. So every printer has a different range of sterilization, various techniques of sterilization it is compatible with. Every material has various techniques it is compatible with. Short note about biocompatibility. If you're implanting something or if anything is likely to come in contact with patient, you need to pay attention to biocompatibility, which is intrinsic to a particular material and slightly affected by the technology that you're using. So what is the adoption path? If you want to go back home today and start 3D printing, you will need to form a team, a surgeon, radiologist, OT technicians. The surgeon and radiologist will have to learn basics of segmentation. It is basically just drawing. OT technicians will have to learn very, very basics of CAD, CAM, and 3D printing, which are at a textbook level or few instruction videos available online. Your radiologist will have to tweak routine imaging protocols because we don't want to expose patients to radiologic imaging just for virtual planning or 3D printing. So your routine, pick one golden easy use case, start with anatomy models, refine with repeated practice, move on to sterilizable surgical guides. You can also do more advanced cases which you keep on outsourcing. So actual implants and prosthesis you outsource. When your printer costs 2 to 3 crore, you want to outsource that case to someone who has that printer. Track your outcomes from day one. So you can track these metrics over time: blood loss, warm ischemia, complications. Safety QA and regulatory, I'll be sharing this PPT so you can go through these rules and you will see, it is not that complex. Potential pitfalls, so it can be frustrating and time consuming in the beginning, needs patience and practice. AI solutions are there, but they're trained on adults. In pediatrics, they might go disastrously wrong. Soft tissue drift might happen intra-op, so you need a backup conventional plan. Evidence. So thank you. If you scan this QR code, you can download this presentation. You can please feel free to reach out. I'll be glad to take any questions.
Thank you, Dr. Patel. Any questions, please, from the audience?
So if you have any radiologists in the audience, no. So every CT scanner takes a particular CT scan with a acquisition thickness. There is a way, if you've written the raw image in the CT scanner, there is a way to reconstruct it later with a thinner thickness also. So for my clinical workflow, I always reconstruct things with a 1 mm thickness.
1 mm.
Yeah. So even if it is 5 mm, you can then reconstruct it as long as the raw data is saved in the computer. So if it is an in-house scan, that is usually possible. So 3D printers will print in layers that are in 0.25 mm, 0.2 mm, 0.1 mm. So the thinner your CT scan is, the better the data acquisition for the 3D printing. No, so the slice count.
Slice count.
Yeah, as long as your patient is still, it doesn't
No,
matter.
no. What I want to say is the CT scan, the capacity will be 56 scan or 512. That different scan 64. So what is the ideal one for this 3D?
So the larger the slice count, the less motion artifacts you have. So if your anatomy is still, it doesn't really matter. So our department doesn't need something that is so fast. We have a 32 slice CT scanner.
32 slice.
So that is also enough if the patient is still, the anatomy is still. So for something like respiratory motion that is inadequate, there 256 slice minimum would be required.
Thank you, Dr. Nirad.
Thank you.
We'll move to the next session. Next talk.
Next presentation is by Dr. Sujata Mohanty. She's professor and head of Department of Stem Cell Research Unit in AIIMS, New Delhi. She'll be joining us online.
Very good afternoon to everyone. My slides are available.
Yes, ma'am. Your slides are visible and you are audible. Please.
Thank you so much. Thank you so much. Very grateful to the organizers. And my topic is 3D bioprinting, bridging lab to clinics. So I think the previous speaker, Dr. Desai, has made my talk very good. The only difference is when we're talking about bioprinting, you can see here we have a dedicated place to work on stem cells and tissue engineering, where 3D bioprinting is one of the aspects we are just initiating. And it means we are working on stem cells and also stem cell derived intracellular vesicle, which are very small particles as compared to others. They are close to virus. They are having the same coordinate. This is very thing is number one. We also do electrospinning, which is you can make thin sheets, and also also the 3D bioprinting, not different from 3D printing, except that you have to include some of the growth factors and stems. Rest of the procedure is same, but bioprinting is very important here. 3D printing, it can be some organic material, but for 3D bioprinting, the ink should be able to support the stems. First example was in I think the was trachea from a bone trachea, the liquid of the cartilage ring, their mechanical properties. So we have three different prototypes. You can see here it is cylindrical, spiral, and bell shaped. First tested their mechanical properties. The procedure how this trachea was printed on the 3D, like tube-like structures. They have a different shape, and this is an electron microscope, the different layers. So we check the mechanical properties so that the trachea does not collapse under the neck movement or dizzying or coughing. And will it be able to support the cells? So we use mesenchymal stem cells, and we found that this of the prototype, the fellow shape was much better in terms of mechanical properties and also very compatible with the cells. So we also did the compatibility, like it should not cause RBC coagulation, which is a very mandatory for any scaffold to be transplanted in humans, which will come in contact with the blood. So hemocompatibility wise, also this material was perfectly fine. Also coated this trachea with agitated gelatin, which will give us much more biocompatibility. It will attract cells and again it is helping the mesenchymal stem cells to differentiate into monocytes. These are the initial findings, and we are doing animal studies now. Second example is the spin. So we are using 3D bioprinting. We started with different shapes and different material. So our first thought was, let us go for the normal bioprinting. Commercial buying is very costly. So Dr. Amushi in my lab, what she did stem cell derived conditioned media. I'll not go into the detail. Everything, mix them together, and she made bioprint. We also tested them in vitro and also in animal model of movement. You can see here that this non-biotic has the capability to make different structures. That means it is not running like water or it is not solid. Show you one video where you can see that this bio is having a good control. You can have multiple shapes and sizes, multiple layers, and you can have different type of cells housed. Uh, so this scaffold is having so for the exchange of media inside the scaffold, so that cells which are inside the scaffold do not die. They get a lot of nutrient and also the oxygen to survive. The 3D image of the bioink with different cells. You can see here, these are the cells, and this is like the cytokeratin or the cytoskeleton of the cells is contained. Blue is the nucleus, and the green is the cytoskeleton. She did choose two different type of cells to test her bioink. The three different skin fibroblasts and skin keratinocytes. You can see there is an increase in both keratinocytes, fibroblast, and day 14 under this new bioink, which is called Gelma HM. Increase very nice. Both fibroblast. And these two type of cells has good migration because healing is by the migration of fibroblast and keratinocytes. You can see here, 36 hour, this normal biochemistry has shown the maximum migration of cells. This is fibroblast and this is keratinocyte. So in the animal model of bone injury, I would like to focus on the last panel, which is our test group, Telma and the normal bioweek. The healing is perfectly fine and much significant from day 7 onwards. And day 10 is complete healing. Commercial product, the Apollo sheet, it heals the bone approximately around 14 weeks. This was a very promising finding, and we are moving ahead with higher. These are all pathological staining also confirm the same thing. We made the bioink, it is under patenting, it is easily formulated, it is economical, and it has shown better bioink. Second is like in 3D printing, we are going for the core. So why core is important? Our aim was to have dual loop. If you 3D print, the printing technique creates one core between two lines, for one, and single line like pencil. That's why we got dual. We did it by incorporating and HCl. So it shows that yes, by the particle all green means cells are right. Red dot means there are some cells, but I is by the particle bone mineralization, which is indicated by CN labeling and collagen 1 labeling, shows that this scaffold osteo so apart from 3D printing, we also go for other fabrication methods. So I would like to touch upon that. As I said that we were using amniotic membrane for transplanting liver epithelial stem cells. It was very good, very bioactive. But we used electrospinning technique to make a scaffold. So why the need is the electrospinning is like a thin stream of chemical is going on, collected on the drum, and then you have an emission. So the requirement was this is a electronic membrane like the OT. It is very difficult for the surgeons to pick up because when it is wet, it behaves like a wet tissue paper. Very difficult to spread over either over the skin or over the eye. And sometimes when there is heavy manipulation, it tears. Keeping that in mind, we use electrospinning, make a scaffold, sample scaffold, which is very easy for maneuvering. You can see here, semiotic membrane with electrospun PCL nano sheet. Very easy to handle. You can spread it around very nicely, and it has the same supportive ability for the skin and the eye stems. So this is the technique, like she did electrospinning membrane and then biotic membrane. She did the compression, tested in animal model, and it has shown very good antibacterial, prostatic, anti-inflammatory, and to promote angiogenesis and epithelialization in the skin model. This is what the membrane look like under the electron microscopy. This semiotic membrane with PCL membrane sandwich here. This transparent in animal model again you can see here shows better and also histopathological examiner says that better neoepithelialization, profound collagen deposition, generation in the skin. So from cell, I will come to cell 3. As I told you, that stem cells secrete some amino acid, which contains cytokine growth factors. So these are, they don't have any nucleus. So a cell needs to be viable for its activity because it is replaced, it is dead. Most of the properties are gone. But these extracellular vesicles, because they do not have a nucleus, their shelf life is more, but they have the same properties. Their size is equivalent to any virus particle. So, and they are vesicles. So my PhD students should be start blending different material like synthetic protein and protein membrane and external vesicle from the stem cells to make scaffold. She named it as a she, and she has already got the patent for the scaffold. So this was used by freeze drying method, like just mix together all ingredient, keep it minus 30 or minus 80. And after some time, we just dry them. And when there's ice crystal, then they evaporate. They leave some space at the start. That would become very spongy and very flexible. See here, it is very spongy and flexible, and can take the shape of any mold which is there where you want to place it. This is again was tested in the one model. So this is also showing remarkable wound closure capability from day 7 onwards when we geoderm has a control, which is again a commercial one and very costly one, was showing some great 3D properties. Again, these are all histopathological analysis which are the most standard. So now what we are doing is we are going for the organoid. These are the complex cellular structure in culture. We are going to have this technology for screening of drugs, different organoid from liver, brain, and the intestinal. This is just showing that how small single droplet will contain a group of organoids like that for drug screening. Now, what are the innovations? Because they keep you driving for achieving more depending upon the requirement. Handheld 3D is coming. They have this cassette or syringes which can be filled with biotic cells. Cartilage generation done, surgery done at the OT. After some time, you can see the cartilage is being and the defect is being reduced from the size. This is one of the interesting paper which I liked. And another one is like people are going for full thickness skin and they are comparing it in terms of mixing together all types of skin cells like melanocytes. Like you have skin substitute which is matching as per the individual's skin tone. Here and adipose maintain the plasticity, the thickness. So they have mixed different type of bioing, different type of cells, and that has resulted in a full 3D printed skin. And this skin has shown better regeneration in the swine model. You can see this 3D printed skin pathology or immunocytochemistry data as compared to the normal human skin, it is very closely matched. So, and I would like to say the take-home message would be it is very promising, but we need to do the scaling up. If we do not do the scaling up, then it doesn't meet the patient's requirement. Also, we should keep in mind the patient-specific scaffold. So it is not one size fits all. There are different age group, pediatric versus adult, and also different sizes. So we need to cater to them, which make it 3D printing has the upper hand. We need to understand the 3D microenvironment of cells. I talked to you about one type of cells, the cartilage or bone. But when we see in vivo, it is always a crosstalk between different type of cells that we saw in the thickness skin, 3D printed skin. We need to understand the crosstalk between the different type of cells and bring them together in the finding and to mimic the native tissue. These are the things which needs attention and more streamlined. And I would like to acknowledge some of the students which I mentioned here in the presentation, but also others who have taken part here. Whole AIMS facility and Dr. Radhika Tandon from ophthalmology, Dr. Sumesh from the dermatology, Dr. Ashwini Agrawal and Dr. Begunajit Basu. They were my collaborators both from IIT Delhi and IIT Bangalore, and also the funding agency for their generous help. So thank you all. I would like to bring to your notice that we have an Indian Society for Extracellular Vesicle, which will be having their second annual meeting, which is for 2026. Your city, it is in Bhubaneswar, Institute of Life Sciences. All are most welcome to join us and participate for engaging discussion. Thank you so much. If any question, I'm happy to.
Thank you, ma'am, for a nice talk regarding the biodegradable scaffolding molecule and also the use of the 3D bioprinting. As a pediatric surgeon, I know there are some disease conditions where this technique can be utilized. I may invite some queries or questions from the audience to ma'am. Ma'am, can we utilize the same scaffold for many procedures using the 3D bioprinting, or you have to discard that?
Absolutely. So, as I said, that these scaffolds needs to be tailor-made depending upon the requirement. We are using amniotic membrane based scaffold for when skin is not healing. And some of the surgeons in our use, they have used for pediatric purposes. I think one for spina bifida, and there were some other 2, 3 more procedures where they have used. And also on some skin wound, like burn wound. Some pediatric cases, there were some wounds which was needing immediate anti-inflammation and also regeneration. There we have used amniotic membrane-based binding for them. But they were not 3D printed, but they were freeze-dried.
Thank you, ma'am. Any queries from the audience or my co-chairpersons? Thank you, ma'am.
Thank you so much.
We'll end this session here. Thank you.
Session.